Cool. So I changed the title of my talk to accurately kind of reflect what we're going to be talking about, which is really adventures in exploring some of these multi-ancestry biobanks. And it's going to be actually a little bit of not repeat stuff, but it actually does end up tying back really nicely to the work that we've been doing on repeats in terms of looking at how these behave differently in different ancestries and how we can tie that back to some of the things that we're finding when we start to look at ancestry in these large biobanks. Okay, so I'll start with this picture that kind of really drives a lot of what we've been thinking about in the lab. So as statistical geneticists, we spend a lot of time doing genome-wide association studies and collecting hits that look like these, of different regions of the genome that are associated with some trait that we might be interested in. And I've always been kind of amazed that if we look at the pictures that are coming out of things like GWAS, we found all of these hits, but for the vast majority of these, we actually don't know what are the causal drivers, what genetic variants are actually driving that signal. And we're still not like amazing at turning these pictures into predictive models that can accurately predict somebody's, say, risk for a certain disease. That gets even worse if we look across different ancestries. And this is really great work from Bogdan Pisaniak's group, showing that if you make a risk score to predict somebody's risk for a disease, and you train that on, say, data from European populations, well, the accuracy of those models is really going to decrease when you apply that to populations that are further and further away from the training population, which again is usually European populations. And so what do we need to actually tackle this problem? Well, we need a combination of things. One is that we need to just start looking at more data from a range of different human ancestries. And we also, of course, need to think about methods for how to apply some of these things like GWAS and risk scores and so on to multi-ancestry cohorts. And by the way, I don't know kind of how the seminar series usually works, but feel free to raise your hand and interrupt with questions throughout. I would be happy to have it be a discussion. Okay, on the data front, things are getting better. So we, along with probably many other people, have spent a good part of the last 5 to 10 years looking at data from UK Biobank, which is great. We have 500,000 whole genome sequencing data sets and a bunch of different phenotypes that have been collected. But of course, the limitation of that data set is that it's the vast majority of it is of European ancestry. Now we're really excited to move a lot of our pipelines to the All of Us cohort, which has been coming online. They're now up to about 500,000 individuals with whole genome sequencing data, targeting 1 million eventually. And that data set is much more diverse. It's only about 50% European. And I put the MVP up here as another example. There are many of these multi-ancestry biobanks that are coming online that are presenting kind of new opportunities to look at the signals that we had found previously and see how does that generalize and how does that look when we expand to different populations. But as we started to analyze things like the All of Us cohort, we realized that we actually had some questions for how we should be doing these types of analyses that we want to do. So one kind of thought that we had was, well, we could just take this large cohort like All of Us, and we can just break it up into different ancestry groups. And maybe we can analyze the Europeans and make a model, and we can analyze the African Americans and make a model, and so on. And in fact, that paradigm has given some interesting and useful results. And in fact, if you look at really useful resource released by the All of Us project, they have these all by all tables where they've done all the genotype correlations across the data set. And they've indeed broken it down by these different kind of categories of predicted ancestry for different individuals. However, if we actually go and look at ancestry is ultimately a continuous phenomenon. This is showing a PCA of individuals from the All of Us research project. And the colors here are coloring reference populations, like you might get out of something like 1,000 genomes if you've worked with that. So, African, East Asian, et cetera. And the gray dots are all the individuals in the All of Us cohort. And hopefully you can appreciate that, well, not everybody is falling neatly into one of those buckets. So if I now want to go make a model for, say, a risk score for those different groups of people, well, for a lot of us, it's not clear which bucket or, which model would be most appropriate for us. And one kind of, additional note on how we treat ancestry is, well, should we also take local ancestry into account? A lot of individuals in the All of Us cohort, and in fact, probably most individuals to some degree, are likely admixed between different populations. And that ends up not only at the global ancestry scale, bringing in this diversity and this continuum of ancestry, But it also, if we look at the local ancestry scale, so we'll zoom in on individual regions of the genome and ask, okay, at this position of interest, do you have a European chunk, or do you have an African chunk or an East Asian chunk, and so on at that individual position? And in fact, different individuals that have the same global ancestry, more or less, might have very different pictures when we look at the local ancestry. And actually, we wrote a whole grant about, oh, let's incorporate local ancestry into risk scores and GWAS. And then, as I'll talk about, we kind of take a step back and say, well, does that actually even make any sense? And when would we actually even want to do that? So back to kind of the strategies that we've been thinking about for how to deal with analyses in these large cohorts. One question that I brought up in the beginning that we've been struggling with is to group or not to group. When I go and analyze something like this all of us cohort, should we break it up into maybe genetically similar subgroups and make different models for each of those subgroups? Or maybe it's actually a better strategy to just ignore groups and ancestry labels altogether and just do analyses across the entire cohort at once. And we realized that we didn't actually know how to answer that question because the optimal strategy likely depends on how much of the genetic architecture of whatever trait we're interested in is shared versus ancestry specific across different groups. And as in kind of anything in biology, the answer is probably, well, it depends, right? Some traits are probably largely shared. This is just looking at Manhattan plots for LDL cholesterol in the all of us cohort, looking at either individuals with African or not African ancestry. And there's some differences, but more or less the signals that we're seeing for LDL cholesterol are kind of largely falling in the same places. On the other hand, here's a different example. This is white blood cell count in the all of us. Again, looking at a Manhattan plot for these two different groups. And there's some pretty crazy differences, right? There's this huge spike on chromosome 1 for the African samples. This is driven by the Duffy blood group region, which is well known to be very differentiated between European and African populations. Has a huge effect in the admixed Africans, but there's just no signal because it's not really polymorphic in the Europeans. And then there's this other signal hanging out here on chromosome 17 that wasn't found in the African individuals. Right, so in some cases, the genetic architecture of the trait might actually look really different. So this kind of questions helped us narrow down on some more specific things that we thought we had to answer before we could go back and do these analyses in these cohorts. And this is going to motivate the first kind of set of questions that I'll talk about. So we realized that we wanted to try and answer this question of, are the genetic effects for these different traits that we might be interested in, are they shared or ancestry specific? On one hand, we might have signals that look like this. Maybe there's a variant. Here's an A to T SNP. It's maybe polymorphic in two different populations that we're looking at, and it has a similar effect in each of them. So that's an example of a signal that we would say is shared across groups. But maybe there's also signals that aren't shared across groups. And what might those look like? And those can actually come in a couple different flavors. Here's one of the flavors is that we have a variant. It's polymorphic in one population and has an effect, but that variant is just missing from another population that we're looking at. So there's just no chance for it to have an effect. So this is kind of like a trivial way that something can have an ancestry-specific signal. Here's a second way that's a little bit more puzzling for how something could end up showing up as ancestry-specific. Maybe there's a variant that has an effect on a trait of interest in one population, and that variant is polymorphic in the second population, but it's not associated with the trait. It's not actually driving the trait. And so questions that we had are, well, how often do these cases actually occur? And if these things like these ancestry-specific signals are real, then can we actually detect specific examples and try to understand what is happening at those? If they are ancestry-specific, are they due to global ancestry, local ancestry, something else? And if we had a better picture of what's going on for these different cases, could we use that to improve risk score portability, our ability to transfer things like polygenic risk scores across different populations? Okay, so that was kind of motivation to bring to kind of three different projects that I'll talk about trying to get at this question. And we'll start with this first question of, can we find specific examples of effects for complex traits that look like they are ancestry specific? And this is really great work that is led by Tara and Wilfredo, who are two graduate students in the group. Okay, so Tara's project was to try and get at this using something called fine mapping. And if you're not familiar with fine mapping, the idea of fine mapping is, well, I've done a genome-wide association study. I found all of these signals for my trait of interest. But actually, the majority of the variants, each dot here is a variant, that are associated with GWAS are not actually causal variants. They're just correlated with something nearby. So the idea of fine mapping is you can zoom in on these hits in your Manhattan plot. You can combine that with other information like a reference panel that tells you about the correlation between different variant types. And then you can use that to compute a causal probability, or we call it a PIP here, giving you the probability that each variant in your data set is actually causally driving that signal. So that's fine mapping. You can do kind of an advance on the single study fine mapping by doing multi-ancestry fine mapping. And the idea here is that if I've done a GWAS for a trait that I'm interested in multiple different populations, and they have different LD, so different pairwise correlation between the variants in the region, well, sometimes you can use that to improve the resolution of fine mapping. So maybe two variants were super highly correlated in European population, so you couldn't tell which was causal versus just correlated. But then you go to the African population and that correlation is broken down. You can actually infer which one was more likely to be the causal variant. So multi-ancestry fine mapping, you can similarly get out this causal probability at the end. So Tara asked the question, well, if I can do multi-ancestry fine mapping and actually figure out what the causal variants are, if I know what they are, then maybe I can use that to help me answer this question of, well, of those causal variants that I found, which appear to be shared across ancestries versus acting in an ancestry-specific way. And we really drew kind of inspiration, actually heavily from the MS Caviar paper developed here at UCLA that did one of the early versions of multi-ancestry fine mapping. And it goes something like this, where you've done GWAS, maybe in two different populations. You have the effect sizes and the p-values from your GWAS, and you also have a reference panel that tells you about the pairwise LD, or the pairwise correlation between all the different variants. And then we can write down what's called these different causal configurations. So I can ask for each different variant. In this case, I had 4 variants. I can write down these configurations where I say each variant is either not causal, 0, or causal, with a 1. So here I just wrote down 3 different potential causal configurations for which variants are causal versus not. There would be, 2 to the n different causal configurations that you could write down here. And then you can combine this with a prior probability of each of these configurations, and you can use this to compute the posterior probability of each possible configuration, and then use that to compute the causal probability of each variant in your data set. So if I wanted to know the probability that this variant RS1 was causal, well, then I could add up the contribution from all of the configurations where that was set to 1, and so on for the second variant here. And this works really great for kind of breaking down that LD that can be different between different populations. But this kind of framework, by definition, is looking for shared effects. We only had 1 causal configuration. It just had a 0 or 1 for each of the possible variants as being causal or not causal. And we really want to know about effects that are, ancestry specific versus shared. So we realized we wanted to extend this a little bit to try and get at that. So Tara's project was to develop this method called PipSort that tries to take that original framework and now extend it to find either shared or ancestry-specific effects with the same model. And she made just a couple of modifications to this framework. One is that her framework can handle variants that are present, say, only in one study and not in another. And if you remember back to the ways that we talked about ways that things could be ancestry specific, well, one way is just that variant is present in one population, but missing from the other one. So that's what these NAs here are, things that are missing in one of the groups. And the other thing that she did was take those configurations that we are examining, and now instead of having a single vector of 0s and 1s for causal, not causal, she just extended that to have population-specific causal vectors. So now we have, a number here for each of the different populations saying for this particular variant, it's either not causal or 0, or causal or 1, or missing is the ones that are the X's. And then we can similarly bring in a prior probability of each configuration. I didn't talk about this for the previous slide because it maybe didn't matter a whole lot. But here we had to bring in a prior. And the key part of the prior was asking what's our prior expectation that a particular signal is shared across populations. And if we set that prior to something really high, then maybe we would upweight configurations like this one, where the causal variant is shared across studies, and downweight ones where it's not shared. And in the opposite, if we set that prior probability to something low, then we would upweight things where the causal variants are not shared. And then we can, similar to what we did in the previous method, if we wanted to find the posterior probability that any particular variant is causal, we can just add up the probabilities of the relevant configurations. The key difference here is that because we have now study-specific configurations, we can get study-specific probabilities of causality. So if I want to know the probability that this particular variant is causal in population 1, I can add up the contribution of the configurations where I had a 1 for that variant for population 1, and so on to get a posterior probability that's specific for population 2. So instead of having one probability of causality across everybody, we now have a probability for population 1 and a separate probability for population 2. Kirk has a question.
How are the X's handled there?
So they're not actually X's, that is just missing from the matrix.
Yeah, so how are the population-specific ones handled in the PIP scores?
Oh, well, they won't, I guess they won't have a probability for the population where they're missing.
Got it.
Yes. Good question. Okay, so yeah, just a side note on this. So we get these two different probabilities for each variant, one for each of the two different populations. The number of configurations, it already exploded with MS Caviar, where you could have 2 to the n possible configurations. Now we made it even worse because we like doubled the size of the configurations and the dimensions of all the matrices double. And so we've had to do some computational tricks to make this better. So one is to limit the total number of possible causal variants. And we also now are implementing shotgun stochastic search so that we don't have to explore like every possible configuration. So more of an approximation approach. Okay, we've done a bunch of simulations to see how PipSort behaves in different scenarios. I want to really get to the real data, so I'm not going to spend a lot of time on simulations. I just wanted to show one simulation set to illustrate a point about population size imbalances and the impact of this sharing parameter that I mentioned. And before I get to what's here, so we did multiple simulations of things that are either shared or ancestry specific. The summary for shared things is that they're pretty easy to find. If it's shared with similar effect across multiple populations, all the tools that we try get it pretty well. Where the real challenge comes in is looking for things that are ancestry specific. So here's one example simulation that we've done where here's a variant that we have a single variant that is causal in Africans and not causal in Europeans. And then we looked at either the European PIP or the African PIP, so those posterior probabilities of causality for each variant. And the red dots are the African cohort, and this is UK Biobank. So note the population size imbalance. We have 330 European samples and 7,000 African samples. But in any case, even with this sample size, we can get it right in the African samples. And then we looked at a European cohort of matched sample size of 7,000 samples, and we looked at the PIP for that variant in Europeans, and we saw it basically at this sample size followed the sharing parameter, right? So it was really, we didn't really get a whole lot of information. It was really hard for us to distinguish that this was not causal in the Europeans. Only after we upped the sample size to the full available cohort of European samples could we start to accurately distinguish that, okay, this is causal in the African samples, and now I have enough data in the Europeans to say it's not causal there. And so this is, really, for us, it was informative about, the impact of that sharing parameter. And, really at small sample sizes, the prior really matters. And once you get to a big enough data set, the prior, as it should, doesn't matter quite as much. But it also highlights the challenges that we still deal with, even when we go to all of us, with this huge imbalance of the sample sizes across the different populations. So even though we can develop cool methods to look at these things, we still need to get more data than the other populations. Okay, So I want to get now to the real data. So we applied PipSort to two different biobanks. One was the UK biobank, where I just talked about the sample sizes that we have available for these two groups there. And then we also applied PipSort to data from the All of Us, which is that more diverse data set that I mentioned. This actually highlights a challenge that we have with defining the ancestry groups in All of Us. And there we looked at an African versus not African cohort because we found that we had more power if we just grouped all the non-African samples together. But it's important to note that the African cohort in All of Us is quite a bit bigger, about 3 times bigger than what we have in the UK biobank. And then we applied PipSort to 2 different traits in this case, so LDL cholesterol and platelet count. We've now run it on other blood counts, blood traits as well. I'll get to the results in a second, but I just wanted to put a PS that it becomes really frustratingly difficult to compare results for these big analyses across different biobanks. And I'll go ahead and say it's like a miracle when fine mapping results replicate for individual loci across these different cohorts. There's all kind of challenges that came up with, maybe the variant filtering procedures are different in one versus the other. For sure, the ancestry definitions of, say, European versus African are, quite different in UK Biobank versus All of Us. So it was an interesting exercise in the real data to actually get the results to match up. One was an HD19, the other was HD38 when we did this analysis. So a lot of interesting kind of technical challenges. Okay, to the real data. So we applied this to a lot of different loci that come up for those different traits. The take home number 1 is that most causal variants seem like they're shared. Most of the loci that we looked at looked like something like this. This red variant here, PipSort prioritized as a high confidence causal variant, and both the different biobanks and both the different cohorts that we looked at in each. And something like 88 to 99% of the regions that we analyzed across different traits look like they have shared signals that are present in both the populations and usually have at least evidence of replication across the different biobanks. So most of the things are shared, but the examples where they're not shared are also really interesting to dig into. Some signals are ancestry specific, and here's an example of one ancestry specific signal. And it gets kind of a little complicated because there's multiple different causal variants going on. This is a locus that's associated with platelet count. But I'll draw a box around one group of variants that was only coming up as a credible set associated with this trait and the African samples from each of the different cohorts. When we zoom in on this set of variants that was ranked highly by PipSort in All of Us, it's these 4 variants here. One of those is a known causal missense variant in this gene MPL, which is a known receptor that's relevant for platelet count. This was a cool example. We found this known causal variant. Why was this ancestry specific? Well, the reason it's ancestry specific is just because it's not in Europeans. This one is 5% minor allele frequency in the African cohort. It's basically not present in the European cohort. So that's why we identified it as being ancestry specific. And in fact, most of the high confidence ancestry specific things that we, and I'd say other people have found, are in this category, where it's just a minor allele frequency difference, and that's why we found it in one versus the other. Interestingly, many of the examples that we found for this type of thing are coding or things where it was actually interpretable what the variant was doing, which is not always the case for the shared signals. But most of these ancestry-specific ones look like this. But what about those other examples? And this is going to kind of transition to the next part of the talk. We did find examples of ancestry-specific things that we couldn't explain by those frequency differences alone. Here's one example. This is a region next to this gene HDAC7, again, associated with platelet count. PipSort prioritized several variants across the 2 different cohorts and the European samples that were highly associated with the trait and it thought were causal. Those variants are actually present and at similar allele frequencies in the African cohort, but there's like radio silence in the signal. And even at things that we thought that we should have power to detect a signal. Here's another example. This is again something for platelet count in the DLQ1 region. There are variants that PipSort prioritized as being likely causal in the European samples. Those variants are present and they should be associated in the African cohort, but we don't see any association. And so this kind of transitions to the next set of things is, well, these are kind of what we set out to find because we wanted to know, do these actually exist? We found some. They are pretty hard to find. Second questions that we have, are these real? Are these actually examples of variants that are present in both but only have an effect in one? And what might actually be driving these types of signals? Why would these exist? One more thing that we did with PipSort to try and start to ask what might be driving these is say, okay, we found a signal that is only showing up in one ancestry and not another. If it's ancestry specific, is that due to someone's global ancestry, just generally being European, makes that variant have an effect? Or is it driven by local ancestry? So as long as, say, you have European ancestry at this region of the genome, then it's going to have an effect. And so we tried to do this kind of stratified regressions to try and figure this out. I first show it as a cartoon because it gets kind of confusing, but we took each of these ancestry-specific effects that we found, and then we stratified our cohorts both based on the global ancestry that we had done in the past, but also by the local ancestry. And then we reasoned if the effect was driven by local ancestry, then even if you have, say, African global ancestry, as long as you had a European haplotype or a European ancestry at that region, then we should still see an effect of the variant that we're interested in. On the other hand, if the signal is driven by interaction with global ancestry or some other thing like diet or environment or whatever, regardless of the local ancestry that you have at that region, as long as you have European global ancestry, then we would expect to see an effect. So we tried to use this to see if we could figure out for some of these regions, which of these two cases it might be. Okay, so I'll show it for two of these cases. So the first was that HDAC7 locus I mentioned, where again, we stratified people either by their local or their global ancestry. And there's a lot of dots here, but the local ancestry at the bottom is homozygous for European local ancestry. The middle here is heterozygous, and the top is homozygous for a local ancestry that's not European. And at this locus, we found that as long as you had European local ancestry, even if you had been labeled as an African sample or an African from a different ancestry, we saw a significant effect. So that was suggesting to us that this particular signal is driven by something with local ancestry. They didn't all look like this. Here's that second locus that I showed, where again, we did the same stratification by local ancestry. And we found that Europeans across the board, even if they had a different local ancestry that wasn't labeled as Europeans, really it seemed like the global ancestry of just European global ancestry is what was driving this to be an ancestry-specific effect. So we found examples that look like they could be either of these. And of course, going forward and going to the next section as well, what would actually cause that to happen? I'll just say a quick note about Wilfredo's project. So Wilfredo and the lab is trying to now do this local ancestry analysis at a larger scale on all of us. And we just released a preprint where he did local ancestry inference using two different methods on the entire all of us V7 cohort. And then we can use actually those local ancestry labels to go and identify different, say, blood phenotypes in this case that are associated with different local ancestries. He found a bunch of signals, including ones that are well known, including things like PCSK9 for LDL cholesterol and some of these hits for Native American local ancestry and different traits. But eventually we're hoping to tie this back to what we found with the PipSort to do just a deeper dive into these signals that seem like they're driven by local ancestry effects. Okay, so this is wrapping up the first question here is, can we find examples of these ancestry-specific effects? And then the answer that we found, which is in line with many other people have shown, is that the majority of effects seem like they're shared across populations. But there are actually really interesting examples that are not shared. In some cases, we have an explanation. It's just the allele frequency. In some cases, we don't actually know why a particular effect is showing up as ancestry specific. Okay, so that is going to bring me to the next part, which is actually going to tie back in a cool way to the repeats that we've been really interested in, is trying to understand what could be driving some of these ancestry specific effects. Okay, so going back to this picture, why might a variant have an effect in one population and not another? We kind of went through different possibilities in the last couple of slides. Maybe there's an interaction with global ancestry, or maybe there's an interaction with local ancestry, or maybe there's an interaction with some other thing like environment or something that is specific to different ancestries. Zooming in on the local ancestry ones. So why might it matter that you have European local ancestry at a specific locus? Why might that determine whether a particular variant has an effect or not? And we really could only come up with two different possible explanations for why local ancestry would even matter. One was maybe there's interaction between different variants. And maybe, here's the causal variant that's driving whatever trait I'm interested in. But population 2 has some other variant over here that interacts with it and makes it look like there's a different effect because there's an interaction between those two variants. The other thing that we came up with is that, well, maybe the local ancestry is not a causal thing itself, but maybe this variant that we found that we thought to be causal in one population, not in the other, is actually just tagging something else that is causal, but that tagging is different in the two groups. So maybe in population 1, this variant we found is just super highly correlated with something else like a repeat that we didn't directly genotype. But in population 2, it's not in high correlation with that other variant. And so it looked like this was only having effect in one population and not the other. Right? And so this brought us back to looking for repeats or other types of not directly genotyped variants that could be driving these types of effects. Okay, before I kind of go on to the data, I just like to introduce what are the types of repeats that we're going to be looking at here. So we've been focusing on two different types of repeats. One is called short tandem repeats, or STRs. These are short, so the repeat unit is between 1 and 6 base pairs, like the CAG repeat shown here. And there's about 1.5 million of these throughout the genome. One of the main reasons that we're interested in repeats, just both in general, but also as a source of variation across different populations, is that they have super duper high mutation rates. So they're constantly kind of mutating and changing the copy number of the repeat, and in some cases, the sequence of the repeat. And that actually can be very different across different populations. We have also looked at these longer types of repeats, so VNTRs or variable number tandem repeats. The main difference is that the repeat unit can be bigger than 6 base pairs. These are also kind of scattered all throughout the genome and have these pretty rapid mutation rates. Okay, before I get I use TRs to refer to STRs and VNTRs together. Before I get to our data, I want to just motivate, this is actually data from Parulo's lab. This is one of my favorite papers, showing that actually some of the strongest GWAS signals that have been kind of dissected to date, end up being driven by things that are not simple, single nucleotide polymorphisms. And some of these explanations can help, explain these ancestry differences that we're seeing at specific signals. And I'll show this one as an example. This is one of my favorite GWAS signals. So one of the strongest GWAS signals for height is over here on chromosome 15 in this gene aggrecan. And it turns out what's driving most of this signal is that there's this polymorphic repeat in the protein coding region of this gene. And if you look at copy number of the repeat versus height, it's just a super strong correlation. And it seems like the repeat is the causal driver of this signal. A second part of this locus that I think is really fascinating is that if you look across different populations, okay, this was like a pretty good GWAS signal for Europeans, but it's the strongest GWAS signal for height in individuals of African ancestry. All right, so here's ACAN over here. And why is that? Well, that's because if you look at the distribution of the copy numbers, it's a pretty narrow distribution in Europeans. But in Africans, there's this much broader and bimodal distribution of the repeat lengths. So this locus actually ends up explaining more variation in height in Africans versus Europeans. It's the same locus, it's the same variant, but it behaves or explains more variance in one population versus another because just the distribution of alleles is very different across different populations. Okay, so one of the goals of our lab from the very beginning actually has been kind of trying to systematically look at the contribution of these types of repeats to different complex traits. And we've spent a lot of work kind of expanding frameworks for GWAS and other complex trait analyses to incorporate these repeats. At the end of the day, it's actually kind of really simple conceptually what we're doing. So in SNP-based GWAS, you're taking each single nucleotide variant and testing, between 0, 1, and 2 copies of the alternate allele and some trait that you're interested in. For repeats, we just, instead of using the zeros, ones, and twos, we look at the length of the repeat at each person, and we do a bunch of regressions between the number of copies of the repeat and whatever trait that we're interested in. I'll show at the end, there's a whole lot of nonlinear examples that we can find. This doesn't capture everything, but most of our tests have been based on these linear correlations between copy numbers and different traits. So Jonathan Margoliash, who used to be a graduate student in our lab, did kind of a heroic effort to set up a pipeline to do this first in the UK Biobank, and now we've ported that to the All of Us cohort as well. In that case, we didn't have direct genotypes of the STRs. We had to impute them with a reference panel into the SNP data. And so he imputed 500,000 STRs into the UK Biobank data. And then for 44 different blood and serum biomarkers, we could do association tests both between all of the repeats in the different phenotypes, but also for the SNPs. Right. Here's example Manhattan plots for just 2 of those different traits that we looked at. This is overlaying the signal of the SNPs and indels in blue with the STRs in orange. One thing we found immediately was that, well, most of the time, the SNPs and the STRs are pointing to the same places of the genome. They are actually mostly in pretty high LD. We can impute most of the STRs from SNPs, so we don't find a whole bunch of new signals. But we found some signals like this, where both of them kind of pointed to the same place in the genome, but the STR was a much better kind of explanation for the signal compared to the SNPs. I want to zoom into a couple of examples to show you some fun things about some of these. So this is one example TR trait association, and it's one of my favorites. So in this example, there's this CGG repeat, and either the 5' UTR or the promoter of this gene, depending on the annotation that you're using. And the plot on the right is showing the relationship between the sum of the copy number for each person, and in this case, their platelet count. But a lot of other platelet counts were also kind of highly correlated here. And you see across many different, this allelic spectrum, this strong kind of consistent correlation between number of copies of the repeat and, in this case, platelet count. This is one example that we found. I'll come back to this example in a minute. Overall, I'm ignoring all the work that we did to incorporate STRs into fine mapping and do detailed fine mapping between the STRs and the SNPs at each locus, which was actually like 90% of the work for that paper and for doing these types of studies in general, was convincing ourselves that these are actually causal. But at the end of the day, we found 118 STR trait associations that passed all of our fine mapping conditions. These include some of the top GWAS signals known for some of the traits that we looked at. For example, I'm not going to go into detail about this signal, but there's a CAG repeat in ApoB that is one of the top signals for LDL that we were able to replicate. This one, interestingly, has some alleles that are only present in Hispanic samples that we've looked at. So this is an example that I'm not dissecting here of one that can be ancestry specific. Overall, we estimated that STRs explain 5 to 10% of the GWAS signals that we did fine mapping for these different traits. As with the PipSort work in the beginning, found that most of these are shared across ancestries. Many of these replicate across ancestries. I don't think I have a slide on this, but the replication is best when we restrict to ones that were competently fine mapped. But as we saw before, a subset of these actually show very different trends across different ancestries, and those are very interesting to take a look at. I'll show 2 examples. So this, going back to that CBL example, the CGG repeat associated with different platelet traits. If we look at the distribution of the copy number of this repeat across the different ancestries present in UK Biobank, there's some pretty notable differences. And I just want to point to that the Black or the African samples are in orange. Those tend to have much shorter copy numbers than the non-African populations. And the colors, don't get too worried about the other colors, all that the non-African populations tend to have longer repeat lengths. And we found is that this locus replicated really competently in all the non-African populations that we considered, but it didn't replicate in the African populations. But then if we go and look at this association, we see actually we did a linear regression to find this, but it doesn't look totally linear. And all the populations that were non-Africans had repeat lengths that were kind of in this range where we saw this linear trend. But the allele lengths that were common in African samples are actually over in this range where we don't really see that strong of association. So this is one kind of cool example of how something can appear to be ancestry specific. Similar to the ACAN example, if the range of lengths is very different across different populations, and especially if it's a nonlinear trend, well, you could have it that, one population has lengths that are in the range where I saw an association, and the other population just has lengths that are outside of that range, and that's why this appears to be an ancestry-specific signal. I'll show one more just cool example of a GWAS signal. This we had found in the UK Biobank, and then we went over to the All of Us data to see if we could replicate this particular signal. And we went over to the European samples in All of Us. We saw a signal similar. I don't have the UK Biobank plot here, but similar to what we saw in UK Biobank. Here, blue is the SNPs and indels, and orange again is the STRs. Then I went to the African samples for this locus in All of Us. And when we looked at just the SNPs, we didn't actually see anything meeting genome-wide significance. And part of that is due to the low power. But then when we added in the STR, we actually did see the signal rise to significance. This actually ties back really nicely to that idea of what could be explaining these ancestry-specific hits that we found, say, with PipSort. In this example, the SNPs suggested that this was stronger in one group versus the other. But it's actually because this repeat is probably the thing that's driving this signal. And this repeat is in strong LD with the nearby SNPs and Europeans, but it's actually in pretty weak LD with the SNPs and the African samples. And so it looked like it was an ancestry-specific effect, but that was just because we hadn't included the causal thing in the analysis. Okay, a couple more things I want to point out for the repeat work. So in the beginning, I kind of motivated this with, can we use this to improve these polygenic scores for things like, disease risk and so on? As a kind of first pass at this, we've been looking at integrating repeats into polygenic scores to predict gene expression. And in this case, we have, expression for all the genes across in this case, this is the Givatis cohort that has lymphoblastoid cell line data. And then for each gene, we can try to build a model to predict expression of this gene using either the SNPs only, so all the SNPs in some window around that gene, or the SNPs and the repeats together. And then we throw those into a lasso model and we ask, do I actually get any improvement when I add the TRs into this model? This is showing a histogram of the improvements that we get in the R squared between the SNP plus STR compared to the SNP only model. Most genes, we don't really get any improvement much, but there's this long tail where we get a pretty substantial improvement in the prediction. I'll just point out one example of these. This is a gene TIM10. It can zoom in and see what were the coefficients that our model gave to the different variants for the different models that we tried. And the top is showing the coefficients for each variant in this region for the SNP only model. And in that case, we gave kind of small weights to a bunch of different variants in the region. And on the bottom is showing the weights that we give to each variant in the SNP plus STR model. And what we found is when we added the STRs into this model, we threw away most of the other variants that we had originally put into this model and replaced it by giving a lot of weight to one particular variant in this region. And when we zoom in on this variant, we see it's a really polymorphic STR that has this super strong relationship between the copy number, in this case of the AT repeat, and the expression of this gene. And that's pretty consistent across different populations. So that was cool that we were able to kind of, what we needed a bunch of different SNPs to explain, we could explain mostly with just this one particular STR. Then the final step is asking, well, does this actually, tying back to that portability problem, does it actually help us make models that are more portable across different populations? Yeah.
On your axis, this is going down, so shorter is lower expression.
Shorter is lower expression.
Do you see a typical relationship between the repeat length and expression? Does it tend to be negative like that?
Yeah, so I'll mention, I'm not going to go into detail. We've done MPRA for these, and also just looking at like GTEx. There's a slight bias towards positive effects like this. It depends on the repeat unit too. Different repeats show different biases.
You have a reason for that? Does it make sense to you?
I mean, that gets to like, what are these STRs doing? And I think there's a lot of different things. The strongest signal we have is like CG-rich things at promoter regions, which tend to show positive correlations. But there's all kind of other explanations. This one is not at the promoter region. It's kind of in the middle of the gene. You know, there's transcription factor binding can be impacted. Some people, that could affect spacing between regulatory elements. It could affect nucleosome positioning. So there's a lot of different things it could be doing. But we do see a bias towards positive effect direction. Cool. Okay, so we took this and then we tried to apply these models to different populations. In this case, we took the SNP-only model and we saw that it worked really great in European samples. And then when we went to the African samples, it didn't really predict gene expression for this gene at all. But then our SNP plus TR model, again, did really well in Europeans, but did a lot better than the SNP-only model. So this is a model that we trained in Europeans, but then applied to different populations. And this is just for one gene. Most genes are not this great, but there are examples like this where, you know, we assume that because we're now adding in what's probably the causal thing rather than just a tag for the causal thing, that then the model then performs much better in the other population because we're not relying on things to say tag this STR in the same way. Okay, so to say one more note on the STRs, getting back actually a little bit to the question. We found a lot of these signals, a lot of them, you know, behave in these interesting ways that are sometimes different across different ancestries. Highlight that we're doing a couple of different efforts to try and kind of validate some of these in a high throughput way. One thing that we've been working really hard on is setting up massively parallel reporter assays to test a lot of these effects. A couple of highlights from that, and then I point you to the preprint for the rest. One, we saw, you know, different repeats behave very different. Some have a bias towards positive, some have a bias towards negative effects. There's overall this bias towards positive effects. And the other cool thing that we found, or one of the other cool things that we found there, is that a lot of these behave in this nonlinear way. Just that in the human data, we just observe like a small slice of possible repeat lengths, and we're doing these linear regressions to find these associations. But in many cases, if we test a big enough range of repeats, you know, it can start to be nonlinear, right? And so in humans, if we had tested this range, maybe this would have shown a positive effect. And if we would have tested this range, it would have shown a negative effect. And so actually what we measure, if we're assuming these linear models, can depend on the range of repeats, which again can be different across different groups that you might look at. The other kind of high throughput way we're trying to go about these is using, you know, these exciting sequence to function models that are now coming out. This is an example with AlphaFold, which I'm not going to go through in detail, but scarily predicts really well our MPRA data. I'm like, maybe we don't have to do the next round of MPRA. But the other thing that we can do, and this is work in collaboration with Anshul Kundaje, is we can start to apply these model interpretation techniques to data either from the MPRA or from the genomic context and understand or try to understand, you know, why is this STR impacting expression and why might this STR in this genomic context impact expression and this other one in this other context doesn't. And maybe we can learn, you know, what features in, say, the flanking regions are actually important for determining that. Okay, getting short on time, so I'll get to part 3 really quickly to summarize that part. You know, for many of these signals that we identify with GWAS, they might be actually driven by these more complex underlying things like repeats is just one example. There are other types of complex variants. And in some cases, these can be ancestry specific, or they can help us explain signals that we thought from just looking at SNPs, we thought they were ancestry specific, but they're actually not. I'll touch really briefly on the third question, which is a little bit of a detour, but was really kind of fascinating to me and informs now the rest of the analyses that we're doing. Is actually just zooming in for a second on non-genetic factors that might be important. Okay, so for all of the association studies that I've been presenting and that we tend to do with things like GWAS or EQTL analyses, we tend to do a lot of linear association tests. where we have some trait that we're interested in, and then we can model it as a linear combination of effects of different variants. Maybe those are SNPs, or maybe those are TRs or something else, plus different covariates. So for instance, it's common to correct for age, sex, and ancestry PCs. But especially for the blood phenotypes that we've been looking at UK Biobank and all of us, and probably for other phenotypes as well, there's a lot of other stuff that can impact these traits besides the genetics, like social determinants, or where you are in the world, or time of day, and so on. And even though these are probably important, we're kind of just like missing them from the models that we're looking at. Here's an example of triglycerides, which is one of the traits that we looked at in both of those cohorts. So this is triglycerides looking at time of day versus triglyceride level stratified by different quintiles of age within the UK Biobank. And we've just been taking the values as is from the test that we got and put them into our GWAS model. But actually, like all this other stuff is like way more variation than what our genetic variants of interest are actually impacting. And so Ross's thought was, well, how can we ignore this? If we deal with this, we can probably get kind of better, cleaner signals in the genetic data. And this work is directly inspired by a paper called Deep Null, which is by Farhad Hormazdiari, who I believe used to be here, that had an early version of doing something like this. And Ross's idea was, okay, both we can incorporate these other types of covariates, maybe time of day, time of year, location from zip code data, but let's also try to incorporate a way to be able to model either interactions or nonlinear effects of those different covariates. So he's kind of taken a page right out of the Deep Null book and just done this with additional covariates, where we can take these different covariates that might be important. We can use your favorite type of model. He used XGBoost or neural network to use these covariates alone to predict the phenotype, and then put that prediction back in to your polygenic score model or to your GWAS just as another linear covariate term. And he's done that and seen if by doing this modeling of these other covariates, if we could improve performance of either GWAS or in this case, polygenic risk scores for these different traits. There's a lot of different colors here. These are just looking at different sets of covariates. And then if we either use that nonlinear null, in this case, an XGBoost versus just a straight up linear model. And the takeaway was that, well, for all of the traits, adding these extra covariates didn't hurt us. Sometimes they didn't do much, but they didn't hurt us. And for some of the traits, I'll just zoom in on a couple down here, adding these other covariates and their interaction or nonlinear effects had a dramatic effect on our ability to predict the trait, with the most dramatic being for vitamin D, but there's other examples as well. I'll just zoom in really briefly on a couple of those. So one of the cool things that we can do with these models is use things like SHAP scores to try and, not just the model improve the prediction, but can we actually interpret that model and see what were the features or what were the interactions that were most important? We uncovered some kind of known ones. So there's this age times sex interaction for LDL cholesterol. So the direction of age versus LDL is inverted for males versus females after a certain age. Similar age times sex interaction for AST. Vitamin D was super crazy. So vitamin D, if you look either at time of year, so across the course of the year, there's this huge fluctuation, or a geographic location, even just within the UK, there were huge sources of variation. And only after controlling for that could we get kind of any meaningful genetic signals. Okay, so I think, I'm about out of time, so I just wrap up that section. So the covariates, that's not about ancestry. They probably do vary across ancestry, but controlling for that stuff can be kind of really important. And then really quick, I'll just conclude. I never got back to this, thought of to group or not to group. What's the optimal strategy? Probably it's going to end up being a hybrid thing. Most things are shared. There's some really interesting cases where they're not. But I would argue that it's actually more important to include all the real causal variants and to deal with these covariate effects are probably more important than maybe sophisticated models of different types of ancestry. Okay, I'll just end and acknowledge all of the wonderful lab members that contributed to this work and our funding, especially the Center for Admixture Science and Technology that funded a lot of the multi-ancestry work. Thank you.